Dr. Madangi Ganapati has obtained her MSc in Genetics and Plant Breeding from Kerala University and PhD from School of Biotechnology, Madurai Kamaraj University. Having a passion for gaining knowledge and for instilling curiosity in fertile minds, she has been active in the field of teaching and research for more than 15 years. Her current research interest includes recombinant protein expression in plants, transgenic plant development and manipulation using molecular techniques, and analysis of changes in basic gene regulatory pathways during transgene expressions, nutraceuticals, and the role in alleviating certain chronic human diseases. She has a number of publications in peer-reviewed international and national journals.
Factors influencing microbial growth. Welcome to the series on food microbiology. This is the second lecture in the part where we are discussing about the intrinsic factors which control microbial growth in food. In the previous lecture, we discussed and saw the role of pH and moisture and its influence on the growth of bacteria. Today we are going to see the other factors which control or influence microbial growth. The first among these is oxidation reduction potential. Microorganisms display varying degrees of sensitivity to the oxidation reduction potential, otherwise called as O/R, of their growth medium. The oxidation reduction potential of a substrate is generally defined as the ease with which the substrate either gains or loses electrons. And when an element or a compound loses electrons, the substrate is supposed to be oxidized, whereas when it gains an electrons, it is supposed to be reduced. So, a good reducing agent will readily give up an electron, a good oxidizing agent will readily take up electron. So, when this electron transfer occurs, that is transfer of electrons from one compound to another, a potential difference gets created, and this difference in the potential or the potential difference can be measured and expressed in millivolts. So, when the more highly organized or oxidized a substance is, the more positive will be its electric potential. The more highly reduced a substance is, the more negative would be its electric potential. And when the concentration of the oxidant and the reductant is equal, a zero potential will exist. And this oxidation reduction potential of a system is expressed by the symbol EH. And when we look into the microbes, the aerobes which require oxygen for growth, they require positive EH values, whereas the anaerobes require the negative EH values or the reduced values. So, now what decides the oxidation reduction potential? Remember, the food, what we call as food, are basically either plant products or animal products. And what we term as food can be the original from the source from the plant or the animal, the fruit, the vegetable, the seed, or the meat or the egg. So, the source of the original food, that is the characteristic of the what is the nature of the plant or what is the nature of the animal from which that food is derived, that characteristic, so that will influence the OR potential of the original food. Second is the Poisson capacity, that is called as the resistance to the change in the potential of the food. The third aspect is the oxygen tension of the atmosphere about the food, and 4 is the access that the atmosphere has to the food. So, here we are saying that there are 4 major aspects. 1, the source, what was the nature of the original food source. 2, what is like when that food get processed, because of the processing changes, what happened to the food as such. 2, third aspect is the oxygen tension. In what type of atmosphere is the food being processed, in what type of atmosphere is it being stored, and then finally the access of the atmosphere, whether it is in a aerobic condition, anaerobic condition, is it in modified atmospheric packaging. So, all this will influence the OR potential of the food concerned. And now we are going to talk about microbes. So, any microbe which now has to go and attack this particular food depends upon as I said, the nature of the food will change, and some bacteria require reduced conditions for growth initiation. Here we talk about the oxidation reduction potential about minus 200 millivolts, whereas others require the positive EH value for growth. For example, anaerobes require reduced conditions for Clostridium, for example Clostridium. The aerobic bacteria and some members of the genus Bacillus require positive. So, when we change this condition, then accordingly the damage to the food caused by those particular food destruction organisms will reduce. And we have this particular class of organisms which we call as microaerophils. These microaerophils will grow slightly better under reduced conditions. Typical example is your lactobacilli. So, let us take now the class, for example, plant juice, any plant based food or the fruit juices, the plant juices. For them, the negative values, the each values is around 300 to 400. And here normally the damage or destruction to the plant, these food or juices is normally by aerobes and moles. If you consider meats, here the oxidation reduction potential would be in the negative values up to minus 200 normal, especially in the minced meat, and in the minced meat around 200 millivolts. So, here the damage is going to be caused by those organisms which can sustain in these conditions. Cheese, these have on the negative values like negative side minus 20 to minus 200, and when organisms grow, the oxygen in the food gets depleted, and as the oxygen gets depleted, you will have further reduction in the OR potential or the EH value. Here the growth is not slowed as the cells make use of the oxygen donating or hydrogen accepting substances in the medium. What we are now trying to see is, as the organism grows, yes, oxygen will get reduced because it is using up the oxygen, but in that particular food substance you will still have components which can give rise to oxygen, liberate oxygen, or there would be components which can accept hydrogen. So, if such components are present in the food, the microbes can still grow utilizing them. So, then what will happen is that the medium will become poorer in oxidizing and richer in the reducing substances as a result of this microbial growth. So, among the naturally occurring nutrients, ascorbic acid, reducing sugars in plants and fruits, and the sulfhydryl group in the meats are of primary importance, where you can which play a role in this. And the presence or absence of these appropriate quantities of these oxidizing reducing agents in the medium would be of very important to the growth and activity of all microorganisms. So, as the microbes grow and the oxygen gets depleted, if the food contains oxygen donating or hydrogen accepting substances, the microbes can still grow. And among natural in the food, among the natural source, you do have appropriate oxidizing and reducing agents. So, their balance, their concentration will actually decide the further growth of the microbe in the particular food concerned. Nutrition content. In order to grow and function, microorganisms require a number of things. Basic is water. They require a source of energy, they require a source of nitrogen, they require vitamins and related growth factors, they require minerals. So, any food or the substrate in which the microbe has to grow would have to have a ratio proportion of these components. Among the microbes, the moles have the lowest requirement, followed by yeast, then gram-negative bacteria and gram-positive bacteria. And the foodborne microorganisms can utilize sugars, alcohols and amino acids as an energy source. So, one or the other, either a sugar or an alcohol or an amino acid is, if it is present, the microbe can utilize that as an energy source. Then complex carbohydrates, for example starch, cellulose, most of the food contain these. These can also serve as energy source, they can be broken down into simple sugars and the organism can assimilate that. Fats can also be used by microorganism as a source of energy. So, irrespective of what is the source of energy, whether it is a sugar or a starch or a alcohol or a fatty acid or a fat, any of these can serve as an energy source for the microorganism. Nitrogen. The primary nitrogen source, in the case of a heterotrophic organism, amino acids are the nitrogen sources. followed by nitrogenous compounds. Some organisms are able to utilize nucleotides and free amino acids, whereas some others use peptides and proteins. Gram-positive bacteria are the least synthetic, and they have to be necessarily supplied with one or any of these compounds before they grow in the food. The gram-negative bacteria and the molds are able to synthesize most of all their requirements, hence they can grow in food having low vitamin B2. Here, what you have to understand that many organisms, if the raw materials for the requirement are present in the food component, most of the organisms, for example, the gram-negative bacteria and the mold, they would be able to synthesize what they require from the nutrient what is present. Whereas, in the case of many gram-positive organisms, they are not able to synthesize most. So, for them to grow, it has to be readily supplied either in the medium, or here in this case, if the medium is the food, in that. So, if the food is rich in those aspects which they want, then the gram-positive organisms can grow. Whereas, in the case of a gram-negative organism, even if one or the other component is absent, the organism would be able to synthesize it from the raw material which is present in the food, utilize it and then grow. Now, compared to fruits and vegetables, fruits have a are lower in B vitamins than meats. They have a low pH and they have a positive EH value. So, usually the spoilage of fruits is by mold rather than bacteria. The next aspect which actually control the growth of microorganisms is antimicrobial compounds. The stability of a food against microbial attack is due to the presence of naturally occurring substances that have antimicrobial activity. For example, some species have essential oils which have antimicrobial activity. The well known among them are the eugenol in cloves, the allicin in garlic, the cinnamic aldehyde and the eugenol in cinnamon, the allyl isothianate in mustard, eugenol and thymol in sage, the carvacrol or the isothymol and the thymol in oregano. These compounds or these essential oils which are naturally present in these plant products can protect against the microbial attack and preserve the particular plant or the plant product from microbial damage. Cow's milk. Cow's milk also contains several antimicrobial substances. The most well known among them is the lactoferrin, the corn-glutenin and lactoperoxidase system. Raw milk also has a rotavirus inhibitor that can inhibit the plaque forming units per milliliter. But this rotavirus inhibitor normally gets destroyed during pasteurization. Egg, another well used food component, it contains lysozyme. Milk also contains lysozyme, and this lysozyme along with conalbumin provides the eggs with a fairly efficient antimicrobial system. The hydrosidamic acid derivatives, the paracoumaric, the ferulic, the caffeic and the chlorogenic acids found in the fruits, vegetables, tea, molasses and other plant sources all show antibacterial and antifungal activity. So, when we look at each food aspect, for example, egg, milk, fruits, vegetables, spices, all of them contain antimicrobial components which are naturally synthesized in them to protect them against the microbial attack. In the case of milk, lactoferrin is an iron binding glycoprotein which inhibits a large number of foodborne bacteria, and it is antagonized by citrate. The ovotransferrin in the egg, which is inhibitory substance in the egg, that it inhibits Salmonella enteritidis, and the cell vacuoles of the cruciferous plants like cabbage, Brussels sprouts, broccoli, etc. contain glucosinolates, which when because of injury or mechanical disruption, they yield isothiocyanate. So, the lactoferrin, which is an iron binding glycoprotein, because of the presence of this, lot of foodborne bacteria do not grow in milk. But if you add citrate or any other preservatives having citrate, this lactoferrin gets inhibited. So, that capacity is lost, then the food is prone to be destroyed or spoiled by the organism. In this glucosinolates are secondary metabolites of plants which are normally synthesized, which are normally seen in this broccoli, turnips, etc. And when they break down because of injury, they yield isothiocyanate. Let us see in detail the lactoperoxidase system in milk. This lactoperoxidase system is that one which naturally occurs in bovine milk. There are three components here: the lactoperoxidase, the thiocyanate, and hydrogen peroxide. And all the three components are required for the antimicrobial effect. Gram-negative psychotrophs like pseudomonads are quite sensitive. So, for the lactoperoxidase system to be active, you require the presence of all the three: the lactoperoxidase, the thiocyanate, and hydrogen peroxide. And each of them are required in specific quantities. The quantity of lactoperoxidase is needed is 0.5 to 1 ppm, and bovine milk normally contains about 30 ppm, which is quite high. And what happens here is the lactoperoxidase system consists of the production of an antibacterial compound from the thiocyanate ion catalyzed by lactoperoxidase in the presence of hydrogen peroxide, meaning when hydrogen peroxide is present in a specified quantity in milk, the enzyme lactoperoxidase get activated and synthesizes an antibacterial compound from the thiocyanate ion, which is already present in the milk. And this particular lactoperoxidase system is not only present in the milk, but it is also present in various body fluids like tears, saliva, gastric juice. The two essential components, namely thiocyanate and hydrogen peroxide, are naturally present in the milk in varying concentrations. The amount of concentration of both the thiocyanate and hydrogen peroxide depends upon the feed given to the organism on the species and breed of the animal concerned. The quantities of thiocyanate and hydrogen peroxide which naturally occur in milk vary, as I said, depending upon the organism, the breed of the organism. For hydrogen peroxide, you require 100 units per ml for the inhibitory system, but natural milk, normal milk contains only 1 to 2 units per milliliter. So, the extra amount has to be provided to the milk to activate the lactoperoxidase system and to preserve the milk. An effective level of thiocyanate is around 0.25 millimole, and whereas in the milk the quantity varies from 0.02 to 0.25 millimole. So, depending upon the breed of the animal from which the milk is taken, thiocyanate level has to be added so as to become effective. When the lactoperoxidase system in raw milk was activated by adding thiocyanate to 0.25 millimolar along with an equimolar amount of hydrogen peroxide, the shelf life was extended to 5 days compared to 48 hours for the controls, meaning normally milk will get fermented within 48 hours, whereas when the amount of the thiocyanate was increased to 0.25 millimole along with an equimolar amount of hydrogen peroxide, the lactoperoxidase system got activated and the shelf life of that particular sample of milk was extended. It did not get spoiled up to 5 days. And the system was more effective at 30 degrees centigrade than 44 degrees centigrade. The antibacterial effect increases with acidity and the cytoplasmic membrane appears to be the target, meaning the cytoplasmic membrane of the organism becomes more amenable, becomes more permeable, and as the acidity increases here, the bacterial membrane will get ruptured, microbes will get destroyed, because of which your milk will get extended shelf life. This picture actually explains what is the lactoperoxidase system. So, the thiocyanate in the presence of hydrogen peroxide ion, lactoperoxidase enzyme gets activated, you get the production of hypothiocyanate, and this presence of this radical will cause disruption of the bacterial membrane, destroy the microbes, thereby protecting the food from microbial spoilage. And this LPS treatment, it does not function as an end product treatment. This is processing, meaning this we can use when the milk is getting processed. So, this will enhance the naturally occurring antibacterial system in raw milk. It is therefore essential that the quality of the raw milk is good, and thus training on milk production hygiene is complementary to the effective application of the lactoperoxidase system of milk preservation. So, here starting from hygiene, how the animals are treated and how they are reared in the dairy farm, to what is the hygienic practice during milk when milk is obtained, to how it is packaged and brought. All this will actually enhance the lactoperoxidase system of the which is naturally present in bovine milk. All food, irrespective of whether it is a plant or an animal food, it is actually a part of a living organism. And because it is a part of a living organism, during the course of evolution, it will adapt or will have some system to fight against microbial infection, meaning the plant or the animal, which actually is the source, would have its own immune system, would have its own efficient methodology to prevent attack of the microbes. In the case of plants or the seeds, if we are talking about, the first of this is what we call as integument. This integument is a physical barrier to infection. For example, the skin, the shell, the husk, the rind, all these is the first barrier against microbial infection. Otherwise, this can be otherwise called as the integument. The structure of the integument also plays a huge role. What is the composition? This the nature of integument, whether it is the skin or the rind, normally it provides an inhospitable environment for the microorganisms by having low water activity, a shortage of readily available nutrients, and also the presence of antimicrobial components such as short chain fatty acids on animal skin or essential oils on plant surfaces. So, if a microbe has to enter, one, there has to be a break, two, there has to be an opening, it has to have a definite water activity. So, if any of these conditions is not met with, then the organism cannot enter and starts causing food spoilage. So, the first barrier which the organism would have adapted would actually prevent the microbe from entering itself, either because of, for example, in the case of animals like the short chain fatty acids, or in the case of plants like your essential oils, the coumarin or the allicin, etc., which are seen naturally. Physical damage to the integument allows the microbial invasion of the underlying nutrient-rich tissue, and damaged fruits and vegetables deteriorate more rapidly than entire product. So, when there is a scratch or a hole or anything damaged on the surface of this outer barrier, the microbe will utilize that opening, gain entry into the inner layer. Once in the inner layer is a rich repository of nutrients. So, there the microbe can happily avail the nutrients and start growing. But as long as the outer integument or the outer barrier was intact, because it is inhospitable, because it is not conducive, the microbes will neither be able to have a foothold nor it will be able to enter into the food. In plants, the injury can rupture storage cells containing essential oils, or it may also bring together an enzyme and a substrate which was separated in the intact tissue. This happens in plants such as mustard, horseradish, watercress, cabbage, etc., where the antimicrobial isocyanates are produced, and in allium species to produce thiosulfonates such as allicin. Here what is you are seeing is a sensitive or evolutionarily important issue. If there is a scratch or if there is an injury or a break, if there is a scratch or an injury or a break, then the plants will release these essential oils by either because of the rupture or because of the enzyme substrate which got separated which was separated in the original tissue, but which got together because of the injury. This in turn will act as a protective layer against microbial growth. The phytoalexins, the second class of antimicrobials which are produced by many plants in response to microbial invasion, for example, the antifungal component phaseolin which is present in beans. The white or the albumin of the enzyme contains the enzyme lysozyme, which catalyzes the hydrolysis of glycosidic linkages in the peptidoglycan layer. The structural polymer, which is responsible for the strength and rigidity of the bacterial cell wall. So, here these two classes, the phytoalexins in the plants and the lysozyme in the hen's egg, actually can go and destroy the bacterial cell wall, thereby causing destruction. And this lysozyme is active against gram positive bacteria, where the gram peptidoglycan layer is much more readily accessible, but it can also gram negatives if the protective outer layer, the peptidoglycan layer is membrane is damaged in some way. Likewise, you have the ovotransferrin in egg white and the lactoferrin in milk, which are proteins which can scavenge iron from the medium. And iron is absolutely essential for all organisms. And if iron cannot be obtained from the nutrient medium, the organism would have evolved to overcome the limitation by producing their own iron binding compounds, which are called as siderophores. These siderophores in turn would absorb the iron which is present in the medium and take it up. The egg white also has cofactor binding proteins such as avidin and ovoflavoprotein, which will sequester biotin and riboflavin, restricting the growth of bacteria, for which these biotin and the riboflavin are essential growth nutrients. In this session, we saw how the naturally occurring antimicrobials, for example, the lysozyme in the egg, the siderophores, the iron binding proteins present in the bacterial cell, all these how they can prevent the microbial attack and help to preserve the food concerned. Thank you.